right, well, welcome everyone. Welcome to the February California Botanical Society seminar. For those of you who are new here, we have a seminar every second Thursday of the month, and you can find the list of speakers on the California Botanical Society website. Tonight we are, excuse my voice, I have a bit of a cold. Tonight we're really delighted to host Dr. Susan Fawcett. Susan is a postdoctoral researcher at the National Tropical Botanical Garden in Kauai, where she is right now, and curator of ferns at the University in Jepson Herbaria at UC Berkeley. She grew up in Michigan. I think I detect an accent. I did my graduate work in Michigan, so I know that accent. And she graduated from the University of Michigan in 2005, from Northern Michigan University with a master's in 2012, and from the University of Vermont with a PhD in 2020. She's also on the faculty at the University of Michigan Biological Station, where she teaches art and botany. And tonight she's going to talk to us about phylogenomics, taxonomy, and deep hybrid speciation, studies in the fern family Thelypteridaceae. And if you can hold your questions to the end, we will have a Q&A at the end of her talk.
Thanks, Kathleen. All right, so this is a family that has been, had a little bit of controversial classifications over the years. Some authors choose to treat all 1,200 species in a single genus, Thelypteris, and with our recently published treatment, it's 37 genera, and there have been some intermediate classifications as well. And so to just give you a little bit of an orientation, there are just a couple species present in California. One of these is Amoropelta nevadensis, which is in the northern latitudes. Usually places that get some snow, higher elevations in the mountains. And Pelazoneuron puberilum variety sonorense. So these are recent name changes in the Jepson e-flora. These both would have been treated in Thelypteris. And this Pelazoneuron puberilum is really in the southern part of the state, and you find it mostly in shady ravines very near the coast. So within the family, there's a lot of morphological variety, especially in leaf division. So some species have simple leaves to up to 3 times divided, but by far the most common leaf division is pinnate pinnatifid, and there's a lot that kind of have that look to them. The veination is really variable too. There's some really important characteristics in terms of how many veins unite below the sinuses, some very regular sort of areoles, more irregular veination. And then there's a lot of genera that have names that are related to the veination: Amblovenatum, Meniciopsis, Goniopteris. A lot of these relate to vein morphology. And then the indument in the family is also really variable and informative. And so these hyaline acicular hairs, or these thin sort of translucent needle-shaped hairs, are characteristic of the family. They're usually unicellular. So transparent unicellular is a really good marker for the family. And then there's some really cool stellate hairs. The genus Goniopteris has star-shaped hairs. And so this is a family that really rewards close observation. Getting it under the microscope to see the details of the veination in the hair is really critical for even generic placement at sometimes. Lots of variation in glands too, from shape, size, distribution, color. Some have really brilliant ruby red glands or yellow glands. And then the spores are just amazing. So I'm just going to take a moment to share some of this diversity, just because it blows me away thinking about pollen. It's easy to tie form to function and thinking about sticking to a bee's leg or drifting on the wind. But it's always so amazing to me that there's so much morphological diversity in spores these things that we just assume are all basically wind dispersed. So these are all members of Thelypteridaceae. So I worked on a study when I was at University of Vermont trying to see if there were some informative patterns across the spore morphology. And we did find a few things that were coherent, like a reticulate pairing or non-reticulate pairing had some phylogenetic signal, but was really hard to make a lot of sense of patterns of morphology of spore surface. But through that, I was building a big phylogeny based on all of the previously published molecular data, almost entirely chloroplast data from some of these earlier studies, and putting together these trees and working with Alan Smith at UC Berkeley, which I began volunteering with him in 2013. And so he really encouraged me to pursue doctoral research and was always wonderful for talking about ideas and trying to find gaps in knowledge of different things to work on. And this is one of those families that was one of the most problematic. So I had the opportunity to participate in the Tridophyte phylogeny group back in 2015 when we were putting together the classification and systematically going through fern taxonomy to try to decide how to treat ferns, and then looking at Thelypteridaceae. 10 of the 30 genera were known to be non-monophyletic or had never been tested. Many of the old world genera were not diagnosed by any morphological synapomorphies, and the taxonomy hadn't really been revisited in about 50 years, and without, for many lineages, without the benefit of molecular phylogenetic data, or very limited molecular data. So this was starting to look like a good project. So a lot of the New World taxonomy had been worked out by Alan Smith and other colleagues in Brazil. But the Old World was still pretty confusing in tropical regions in particular. So Richard Eric Holtum, who is a curator of ferns at Kew, did kind of an amazing job pretty late in life. I think he didn't even begin working on this family until he was maybe 70 years old, but he really focused on it towards the end of his life and just did an amazing job making sense of what had been a really big taxonomic mess. So he named 7 genera and made hundreds of combinations and made this really wonderful classification that was sort of a really nice starting point to be able to test his taxonomic hypotheses and sort of refine the system that he developed based strictly on morphology. So that was the goal for my doctoral research, was to use morphological data, biogeographical data, and then adding in molecular phylogenetics to try to address some of these questions. So some of the challenges with working with the Thelypteridaceae is that many of the centers of diversity are poorly collected. So Papua New Guinea has just amazing collections. Some of them are so old we were never able to get DNA, but just to see there's a huge amount of diversity in some of these places that are hard to get to. There's a lot of really narrow endemics, so things that may occur on a single island or single mountain range. There's some degree of polyploidy and reticulation, not as bad as in some groups, but enough to kind of mix things up and make things a little bit more complicated. Few specimens are complete, so there is a lot of, especially in the Solomon Islands, there's a lot of really big members of this family that may have a leaf that's 6 feet long. And so to get a specimen that's really representative of what does that plant look like, I'm always drawn to these small species or small groups. There's radiation in the Antilles that I really love working with because they just make these beautiful little specimens. The plants are kind of herbarium sheet sized, and you can really get to know them. And some of these were a lot harder to work with because you're looking at a specimen and it's just one tiny portion of a single leaf. And knowing things like how the base of the blade is tapered or the tip of the blade is tapered, what the rhizome looks like, these are all really helpful and necessary for identifying things to species. And that's almost never what herbarium specimens look like. And then you also need microscopic character. So looking at venation and hairs and glands is really critical, even to arrive at the correct genus identification. So when collectors are in the field, if they see one, then they assume that they've already seen it and they probably already collected it. So I think they're under collected because without looking very closely, a lot of them look very alike. So here's just one example with a single genus, Chingia. So there are 24 described species, and before we started working, 8 of these were known only from the type, or the type in one other collection, and only a single species had molecular data published. And then, so leaves 6 feet long. In this case, some species have leaves 12 feet long. There's a species that has stinky, irritating hairs, which is not something I'm familiar with in too many ferns. And then they have ephemeral populations that may only occur in the aftermath of a disturbance and then disappear. So revisiting them from historic locality data would be challenging. And then they grow on really steep mountainous slopes and tropical islands that are difficult to get to. So these are some of the challenges. I think in the genus Sphaerostephanos, there's like about 200 and some species, and there's some 66 that are known only from the type collection or the type in one other collection. And a lot of those aren't digitized. So really challenging group to work with. Might be some other reasons people are afraid of them, but I'm not sure. So here's an example of a new species that was found hiding in plain sight. It had been collected by a couple of different people, but it was just growing along the side of the road in Fiji. And it was both on Taveuni and outside of Suva. And this is one that we collected and saw, and it had just had other names had been applied to it. And once we started working with it more carefully and looking at a lot of specimens, it was clear that it was something new. And so, the bulk of my work, in large part in collaboration with Alan Smith, was proposing a new classification system for Thelypteridaceae. So the first thing is just getting into the herbarium. And so Alan was just a huge help in being able to help direct the sampling design. And because he's been at the herbarium working there for the last more than 50 years and has done something like 200,000 gifts for determination, it's just one of the best collections in the world for ferns to be able to go and know that you have something that is probably recently collected, because a lot of people are sending specimens, and then also that it's likely correctly identified, just because Alan has put so much time and effort into that. So having that level of curation as a starting point was really critical. And then also being able to be thoughtful about trying to include the type species for every taxon and targeting gaps or the most problematic groups. So there was a lot of help with the sampling design there. And then, of course, once you have the molecular data, going back into the herbarium, and that's when things get really fun and start to make the most sense. Also digging deep into literature. So I was working at University of Vermont in the Pringle Herbarium and the Tryon Library. So that was a huge draw to work there, as there's this amazing fern library that was adjacent to the herbarium. So just a wonderful place to do monographic work and just be surrounded by all this old literature. And then the molecular side of things. So I was really happy to have the opportunity to collaborate with Emily Sessa, Gordon Burley on this genealogy of flagellate plants, or GoFlag project. So this is a targeted enrichment approach. And so basically it's identifying regions that are highly conserved, and then of about 120 base pairs. And then you have a big flanking region. So having that conserved region really helps you build phylogenies across a lot of diversity. And then you can get some pretty good resolution from those usually non-coding flanking regions that are highly variable. So we ended up with about 600 tips, representing not quite half of the known diversity in the family. And we were able to sample all 30 genera that were represented in the pteridophyte phylogeny group, and the type species for almost all of those. And then we had 400 nuclear loci, or 408 nuclear loci, with an average of 379. So we were really happy with the data set, and ended up with about 500,000 base pair matrix, and did maximum likelihood and also multi-species coalescent analyses. And so this is, you know, from a taxonomic perspective, this is definitely fertile ground. So this is the phylogeny and the 3 most polyphyletic genera mapped onto the phylogeny, following the generic concept that was recognized by PPG1. So you can see, you know, we've got Pronephrium coming out in about 9 places, and Cristella in 4 places. So these were the groups that I was then really focused on trying to understand. And so it was just really interesting to see Pronephrium, for example, that name had been applied to a lot of these things that looked very superficially similar with, you can kind of see the theme here with once pinnate fronds with more or less entire margins, not too deeply reduced. And for each of these species, or each of these genera, they're all much more closely related to something with that pinnate pinnatophid frond division than they are to each other. So it was really getting in and recognizing, for example, this Grypothrix is so named because it has these hooked hairs that are just really distinctive, but you have to get in with a microscope. And then also some really remarkable examples of convergence. So this is an Abacopterus on the left and a Menicium on the right. So these are really, really distantly related from totally different parts of the tree. And they have just converged on such a similar morphology that it's just kind of shocking. And so this is one of the segregates from Sphaerostephanos. So this is just a small genus with just 2 species that Alan named Strophocolon. And it has these really long creeping rhizomes. This is a collection from Fiji and tends to grow in these really sunny places. And so seeing this in the field, it's like it really does have kind of an ecological niche that is quite different from Sphaerostephanos, and has sort of a unique architecture of the veination of the leaves. This is another one that's endemic to New Zealand and Eastern Australia, Pacao. So this was another Pneumatopterus segregate that's basically a tree fern. And the genus name was taken from the Maori language. So it's kind of interesting. Pacao and Pacao roja roja is basically the wing of a bird. So it reminds me a bit of the word pteris in Greek, which means fern and also feather. So this sort of parallel between birds and ferns in different languages is sort of interesting. And then we also named a genus for Richard Eric Holtum. So Holtumiella was already taken in a Holtumia, I think was an algae, and Copeland, who is the curator of ferns at UC Berkeley before Allen, had named Holtumiella, and Holtum himself lumped it into synonymy, I think, with tinnitus or something. So we thought he should be commemorated with the genus for just kind of making sense out of so much confusion. And this was one of the groups that he'd really made a lot of progress on. And then Amoropelta. So this is one of the California groups that we revisited. And so some of you may remember Amoropelta nevadensis has been treated in Thelypteris, or Parathelipteris is another name. And so this is one of those things where just having a huge herbarium collection and being able to put all these things side by side, and it just really starts to make sense. So we treated Amoropelta, we did expand Amoropelta, and Parathelipteris ended up being paraphyletic. And so some things went into Amoropelta, some things went into a different genus, Coryphopteris. And then we recognize subgenera. So here is one from northern Michigan. This is the sister species of the Californian Amoropelta nevadensis. This is one from Sri Lanka, and here's one from Puerto Rico. So you can really see some basic morphological similarities, and these all have different base chromosome numbers, which is interesting. And so here's the distribution of Amoropelta nevadensis and Noveboracensis. And this is just sort of a tree. So I was interested in looking at the dates from Joel Nitta's work. So he created, it's called the Fern Tree of Life. It's all free, available online. And he just basically continually pulls all the fern molecular data off of GenBank and then generates trees from it. So, and then has a fossil calibrated dated phylogeny. So based on that, this is the sister group of Amoropelta nevadensis and Noveboracensis here. And those two diverged from each other about 7 million years ago. And then this is a mostly neotropical clade of about 200 species that has just been this enormous radiation. And that has happened in about the last 21 million years. So probably there's a lot of different estimates about the age of the Andes and how long that uplift was taking place. But just looking at patterns of diversity throughout the Andes, this group really seems to have diversified during that period of Andean orogeny. And the reason so this is subgenus Amoropelta, and then we called this subgenus Niba, which is from the Anishinaabe-Moan language, which is the indigenous language of people near location of the type species, Amoropelta noveboracensis, and it means to sleep or she sleeps. And so that is the distinguishing characteristic between these two sister clades, is that Amoropelta subgenus Niba, they go dormant during the winter. So they're just up during the summer months and then die back and disappear. Whereas most of these are in tropical climates and are putting leaves out year round. And then these are both old world groups that separated about 27 million years ago from the Western Hemisphere clades. And then this was really fun to revisit. So this is Alan Smith's thesis from his doctoral work in 1969. He published in the University of California Publications in Botany in 1971. He had done a monographic study of what was Thelypteris section Cyclosaurus, which includes what we now call Cristella, and then Palazoneuron, which had been treated as Cristella. And so it was clear for a long time that it probably wasn't Cristella, but the relationships within the family were really not that clear. So it was just really fun and rewarding to pick up where he left off about 50 years ago and to be able to name this new genus and make all these combinations. So this is one of his names, and this is the other genus that occurs in California. So this is these are the damages overall. So 4 of the genera were newly described that are mostly don't have a lot of species. And then these 3 here were all elevated in rank. So they had been treated as subgenera by Holtum. And so we had basically changed the rank. And then 1 old genus was resurrected. And then the rest here in blue were just changes in circumscription. So some of these were expansions or contractions. And some of them were minor, just taking a species or 2 in or out, and some of them were a little bit more involved. So the phylogenetic data was published in Systematic Botany with a lot of collaborators. A lot of people contributed to generating the data and helping with the analyses, generating tissue or sharing their tissue. And then the taxonomic treatment or the generic classification is available for free from support through the Berkeley Libraries. We were able to publish it open access, and Botanical Research Institute of Texas Press If you want to get a hard copy, you can buy a hard copy, or if you just want to download it for free, it's also available. So appreciate the UC Berkeley Library for doing that. And so now I'd like to talk a little bit about Hawaiian ferns. So within Hawaii, there are 12 ferns and 7 genera, and then among these, there are 5 endemic genera, and 2 of these have diversified within the islands. So those are the 2 that I'm most interested in. And the biogeographic, Hawaii is just so amazing for so many different reasons, but from a biogeographic perspective, it's just super fascinating to see where all these different things have come from. So there are some things that seem to have ancestors from East Asia, from Southeast Asia, Polynesia, and then this is Amoropelta, which has the California relative. The whole single Hawaiian species is nested within this radiation of about 200 species that are mostly South and Central American. And there's about 6 species that have made it into various islands of the Pacific and into Africa that are from probably South American origin. And so first I'll look at Monophyletic. And so this is another case where we're being able to have the molecular data and then going back into the herbarium. Everything just makes total sense. So these had been treated in different genera. So the Hawaiian species had been treated in Cristella, and in East Asia, they were treated in Pronephrium. And then you get the herbarium specimens side by side, and they're almost indistinguishable. And so this is really interesting too. One of my colleagues, Nikisha Patel, did a study on Holisticum in the Barrington lab and looking at climatic niche. And she found just this perfect match from some of these habitats at high elevations in the Himalayas. And they'd found habitats in the Hawaiian Islands that were very, very similar. So there may have been some climatic niche pre-adaptation of ferns that had found their niche in montane settings in Hawaii, were able to really take off. But it was just one of those aha moments when we got these specimens side by side. But then the other really interesting thing about Monophyletic in Hawaii is that it has apparently evolved into these other rheophytic forms that are much smaller. So this is kind of a cool move that a lot of fern lineages in Hawaii seem to do, is they have a sort of a large forest understory version and then this really diminutive rheophytic version. So it looks like it may have evolved sort of a rheophytic riverine habit twice. So in this case, this is one from Kauai that's an endemic with a long creeping rhizome. And then this is one that grows on Oahu that has like this really dense erect rhizome and this big mat of roots that cling to rocks when water passes over. So really, really a cool group. And then this is Ho'eokula. So this is again, was another segregate of Pneumatopterus that was very distantly related from all the other species. And there are currently two species recognized in Hawaii, again, sort of a large forest understory habit, and then a second species that is more of a cliffside rheophyte with a much smaller habit. And that the genus name was taken from the Hawaiian language, Ho'eokula, which is the common name for this fern. And so when we finished this analysis, and this had been treated in Pneumatopterus, and saw, okay, it comes out here as sister to this lineage of Leptogramma, Steganogramma, Cyclogramma. It's like, well, cool, we've got a new and Hawaiian endemic Steganogrammoid. And then we sequenced the chloroplast data. And so this was a surprise. It ended up nested within this mostly East Asian genus Leptogramma. So this was interesting. This is the phylogeny, and Leptogramma is just kind of a fascinating genus. Most of the genera in Thelypteridaceae in general, a lot of ferns have some geographic fidelity where they're usually restricted to a single continent. Not always, there are definitely exceptions. But that's the general rule in Thelypteridaceae. And so with Leptogramma, all these blue tips here are all East Asian or Asian. And then we have, there's one that's known from a single river, the Sipsey River Gorge in Alabama, another narrow endemic in Mexico, and then some from South Africa, and then some from the Mediterranean basin. So this really kind of crazy biogeography. And so, you know, we have this Hawaiian thing kind of coming out in this really biogeographically messy and complex clade. And then, so this is another benefit of having the 400 nuclear gene trees, is you can start looking through one by one and seeing. So with that first phylogeny, it didn't matter if it was a multi-species coalescent analysis or doing a concatenated matrix and doing maximum likelihood. With these data, you tend to end up with really high support values, 100% bootstrap support, and the same topology no matter what you do. But then once you start breaking it apart, you can start to see some of the conflict, and also using some of these concordance factor methods to see what might be going on under the hood. So what we found is that Ho'eokula was nested within Menisiopsis and was sister to this Hawaiian group of Menisiopsis. And then in some other trees, it was nested here with a Himalayan group of Leptogramma. So this is interesting because the genus Leptogramma does not occur on the Hawaiian Islands, and there's been no record of that. So seeing it nested within this Himalayan lineage was really interesting. So looking at the morphology, it actually has a lot in common with Leptogramma, but then the sori and the spores are both share something with Menisiopsis, especially the Hawaiian members. So this does look like a case of deep hybrid origin, and coming out where it had in the base of the leptogrammoid clade is there's a Lucinda McDade paper in 1992 examining phylogenetic behavior of hybrids, and that was one of the classic topologies, was coming out as sister to this big lineage that includes one parent. So this is kind of an exciting to see, because these two groups diverged something like 50 million years ago. And so there's some really great new tools now to be able to look a little bit more closely with big nuclear data sets like this. So we were interested in revisiting some of these hypotheses of hybridization and going back into so Alan Smith's work, and he had done some really wonderful chromosomal work. So being able to go back and have a look at some of these hypotheses. So this is an interesting case where Cristella hispidula is a diploid, and it's widespread throughout tropical and subtropical regions of the world. And we included 6 accessions in our data set. And they were all monophyletic. So we know that it is at least one clade that is widespread throughout, basically throughout the tropics, and gets into Florida. This is Pelasgoneuron kunthii, which is mostly in the southeastern United States. It's kind of weedy. It tends to get into greenhouses, and it was recently collected in Southern California as naturalizing. It's naturalizing in Palestine and Israel and in Spain, and it has also been just really expanding its range throughout Alabama. So this is one that seems to be expanding its range. It's a tetraploid. And then this is Pelasgoneuron ovatum, which is another Florida, mostly northern Florida species. So in Alan's thesis work that published 50 years ago, he was doing a lot of careful cytological study. And so he hypothesized hybrid origin because he found what would be like a back cross between kunthii and ovatum. So figured that it was likely sharing a genome with Pelasgoneuron ovatum. So at that point, it wasn't really clear what the generic affinities of these things was. And so it's, you know, it's a little bit messy. It's challenging as a taxonomist of treating, now that, you know, we're seeing that some of these things of intergeneric hybrid origin, especially just a single lineage that hasn't diversified, but is clearly a stable sexually reproducing species. Dealing with that taxonomically can get a little bit messy. So we ended up just following with the plastid parent, which was the majority expressed genome in terms of number of copies, but it's a very imperfect solution. So don't be confused by that. So we were able to look at locus heterozygosity and allele divergence. So this is a really nice way to try to pick out potential hybrids. So we had a lot of hypotheses going in of knowing what it was we wanted to check based on morphology, and in a lot of cases available cytological data to go in and revisit. So what we found is that there were at least 6 different examples of deep or intergeneric hybrid origin within just the crystalloid clade of Thelypteridaceae. So this work isn't published yet, but it's in revision, will hopefully be out this summer. But some of these groups diverged as much as 45 million years ago, and this morphological similarity and convergence has made it pretty challenging to recognize these groups. And I realize this is not the easiest to see exactly what's going on in this figure. But it just really makes me question how common is hybrid speciation in ferns, or how often is this, or has this happened? So that's just something that was very exciting that came out of these data that I'm excited to look into a little more deeply. So I just want to thank Alan Smith for his support and encouragement. Here he is in his natural habitat, which is sitting on a log next to John Strother. And then all of my collaborators, Li Yuang and Yuxin in Taiwan, are co-authors, along with Israel Borokini on that, the hybridization work. And I want to thank Nancy and Amy for inviting me to give a talk in this series in the California Botanical Society, and also to Kathleen for hosting.
Thank you so much, Susan. We can open it up for questions now. Yeah. It's always awkward to clap on Zoom.
Yeah, it's a joke awkward too. I just never know if anybody thinks they're funny, but.
Well, I could start. Oh,
sorry. I'm curious, since like previous to genomics being used, was there just like very few speciation in the fern family, since it's a little bit harder to tell morphologically their differences?
You mean just like from a taxonomic perspective of what?
Yeah, like were there very few lineages of ferns, because it's a lot harder to differentiate them just based off of their morphological differences?
Yeah, it's so our classification, the morphological classification that was recognized by PPG was, I think, was 30 genera. And so we lumped a couple things. We did lump Parathylypterus and Nanothylypterus, but then if you are adhering to a monophyletic species concept, the changes we made were just sort of necessary if you wanted to maintain monophyletic lineages that were recognizable without lumping everything into one big genus. So I think there has been the tendency has been a little bit more splitting, but there's definitely opportunities to split things that are just personal preference. So you can actually get online and see how the sausage is made. With the Pteridophyte phylogeny group has a GitHub page that is open to the public and has all proposals for taxonomic changes in ferns are there, and people can log in and discuss the relative merits of any of these changes. So Actually, Thelypteridaceae was all of the changes that we proposed were voted on last month at the end of January. And fortunately, there was a pretty overwhelming consensus that this people were approving of the changes we made. But I would say, yes, there's definitely been a tendency to recognize more groups through time. And there's some cool papers that actually chart that out. But that's the trend.
That's really interesting. Thank you.
Yeah.
I have a question, Susan. Thanks so much for this very in-depth look at this group of ferns. The question is, do you get a sense that the Thelypterid ferns tend to differ less morphologically than ferns in other families, or that they tend to differ in different characteristics than other families?
I would say they may differ less, but they certainly, there's some groups that you can just look at, from 5 feet away or 10 feet away, and you can just, what species it is. So I think it's this combination of diversity. There's 1,200 species, so it's, up there in the top, 3 or 4 families for species richness. And so I think, having that diversity and then just really needing these fine scale morphological characters to tell them apart. I think that's part of it, but yeah, hard. And then also I think some really similar things can grow in proximity to each other, or it can have overlapping ranges. So I think that's part of it. And then also, because there just hasn't been, until, Alan Smith's made huge strides in working on all these different floras, but I think there really weren't the resources that you needed to key them that were available for some other groups of ferns. So I think people just tended to ignore them or say Thelypteris spa and move on. So it could be a combination. I'm obviously biased, so I'm probably not the best person to ask about how much they differ.
Thanks.
Susan, I have a question. I have some of my students from my botany class here, and we've been learning how to make herbarium specimens. And I was wondering, how does one make an herbarium specimen with a 12-foot leaf?
Oh, so I have my method. So I'll first of all, I'll say, what herbarium is this going to and how much space do they have? So I'm here at the National Tropical Botanical Garden, and they really make an effort to have complete specimens. So it's just like a total joy to come to a place and say, okay, this is a 6-foot long leaf of one of these ferns, and it's mounted on 6 different sheets. And so I can actually lay the whole thing out and like reconstruct what this thing looked like before it was pressed. If you work in an herbarium that has limited space and is starting to fill up and storage is sort of at a premium, first of all, take good notes. And taking notes on rhizome morphology is really important, because in some cases you may not want to kill a fern, or if you have a tree fern or something, you're not going to cut it down to, make a specimen. It just depends. If you're collecting an annual, it's different if you're collecting a long-lived perennial that you might not feel you need to kill. Taking really detailed notes on like the distance between nodes, the nature and size, thickness of the rhizome is critical. Getting a cut as close to the rhizome as you can, so you're getting the scales. The scales on the base of the stipe usually look like the scales on the rhizome. And then if I have a long leaf, I'll just rip all the pinny off of one side, because they are 2-dimensional. And then I'll try to keep the very base of the blade intact. I'll try to keep the apex intact. I'll try to keep, the longest pinna intact. And then I'll just start pruning excess material, and I will fold it back and forth in an accordion style so that it's going to fit on an herbarium sheet and then still capture these most critical blade shape sort of details. And whatever you're not going to be able to capture in a single specimen, and that's my UC and UC JEP's collecting method is if I'm collecting a duplicate, I'll put it on 5 sheets for PTBG and I'll put it on 1 sheet for UC if I can, just because it's, that's a consideration.
Right.
But yeah, just whatever you're not going to capture on the sheet, try to capture in the notes.
Thank you. That's really interesting. Any other questions? If not, I'm going to ask my second question.
Oh, can I ask my second question?
Sure.
Thanks. So, Susan, have you noticed any commonalities in those species that have reached Hawaii? Things that distinguish them from the species that haven't?
Oh, that's a great question. Well, I'll tell you one thing that is totally fascinating about there's a Pseudophagopteris kourodreniana is reached Hawaii and then it basically developed a sort of indeterminate growth pattern and has scandent leaves that climb across the vegetation, which is like nothing else in the genus. So that's kind of crazy. I mean, that's an amazing thing. So it's more the things that the plants do once they get here that is so fascinating to me. And I haven't noticed anything about special about the ferns that make it here. That nothing has stood out to me, but that's a great question. I'll think about that and let you know if I come up with anything. It seems like the fern flora here is pretty well spaced phylogenetically. So yeah, nothing comes to mind on that.
Okay, thanks. For one thing, it would be interesting if somehow their spores tended to be sort of similar in morphology, implying that they really did travel differently depending on their surface characteristics.
Yeah, I think it's we really can't rule out the role of birds in spore dispersal. And the problem with island biogeography and with fern biology in particular is there's the potentiality of a single sporophyte just produces millions of spores. And so, may take only one of those spores to get to an island. So you're just studying these incredibly fleetingly rare events. And it's really hard to track down like, oh, did that come on a bird or in the wind or, so these are always really challenging questions to study.
I was hoping to ask a question about the deep hybridization in the crystalloid clade that you showed. And I was wondering if those are all associated with allopolyploidy, or are there any homoploid hybrid speciation events?
In all the cases that we know of in Thelypteridaceae, it would be hybrid speciation associated with allopolyploidy. Mm-hmm. So I know there's some examples of homoploid hybridization and Cyathea, David Conant's work, but that is definitely the exception. And ferns, yeah, allopolyploidy is the rule for hybrid speciation in ferns.
Are there any other questions? All right. Well, thank you, Susan, so much for your talk tonight. It was fascinating, really beautiful pictures, great illustration of how useful herbarium specimens are. And then I want to invite everyone to join us next month on March 14th. We'll have a talk from Ingrid Jordan Thaden, who's the director of botany, garden and greenhouse at the University of Wisconsin-Madison. So thanks everyone for joining us tonight, and good night.
Thank you.
Thank you.
Thank you.
Aloha. Thanks, Kathleen.
Yeah, thank you. Thank you all.
Thanks, Nancy.